For this upcoming lab exam, we do have two main components. First component is the diagram-based identification questions, and those are going to be representing between 35 to 40 questions. And in which you are going to be shown a diagram related to the urinary system, and you're going to need to be able to identify the different components on that diagram. The second component is going to be the essay question, which has two components, and those are the response of the heart. So one is asking about the response of the heart in regulating the blood pressure, and the response of the kidney to regulate the blood pressure. So those are the two components of the essay question, or they might appear as two separate questions on the exam. One is asking about the kidney response to regulate the blood pressure, and the other one is asking about the heart response to regulate the blood pressure. And those are going to be representing the remaining 10 points for your 10 to 15 points for your lab exam. So you do have 35 to 40 questions. Each one is worth 1 point towards this exam, and 10 to 15 points towards the essay question, or the two essay questions that will appear on your test. And this gonna make a total of 50 points, which is the total points for this upcoming lab exam. So let's start first with the essay question. The essay question is asking about how the kidney regulates the blood pressure and how the heart regulates the blood pressure. When I start thinking about the kidney regulation, I need to remember that the kidney hates low blood pressure, so it responds to a decrease in the blood pressure to increase it back to normal. And that the heart hates having a high blood pressure because it represents like a afterload for it. So in response to a low blood pressure, it wants to reduce it back to normal. So how the kidney acts, if you remember, by activating in case of a decrease in blood pressure, this is going to be detected by the granular cells of the juxtaglomerular apparatus. As they are acting as mechanoreceptors. This detection is by detecting a decrease in the degree of stretch of the afferent arteriole in the nephron. So this tells me that I have a low blood pressure. How would I respond? The granular cells start to release renin. So my question now is how this renin is going to be increasing my blood pressure. So we need to state that renin converts angiotensinogen to angiotensin 1. And that angiotensin 1 needs to be converted into angiotensin 2. This is the active form of the hormone. And this is going to be mediated by an enzyme called angiotensin converting enzyme that is located in the capillary endothelium. Angiotensin 2 will increase my blood pressure by 5 different means. We need to mention them, and those are vasoconstriction, of the arterioles, this results in an increase in my peripheral resistance. And as a result, an increase in my blood pressure. The second effect of angiotensin 2 is mediated, or the second set of effects of angiotensin 2 are going to be mediated by acting on the hypothalamus. And remember, it has two actions on the hypothalamus. First of them is going to be to stimulate the hypothalamic thirst center. And if you remember, the hypothalamic thirst center will give you a thirst sensation that will make you drink a beverage. And when you drink a beverage, this is going to increase your blood volume back to normal. And as a result, you're going to have an increase in your blood pressure back to normal. The second effect on the hypothalamus that the angiotensin 2 is going to be mediated is going to be stimulation of the Adh release. And if you remember, the Adh was responsible to insert aquaporine channels in the collecting ducts of the kidney nephrons. And those aquaporine channels will allow me to move water back from my collecting ducts to my bloodstream. So the water travels freely here back from the kidney tubules, especially collecting ducts here, back to my blood. This allows me to allow water retention. And as a result of retaining water, there will be an increase in my blood volume. And so an increase in my blood pressure will be taking place for me to get a normal blood pressure back. The third effect of angiotensin 2, by which it increases my blood pressure, is going to be to increase the sodium reabsorption. And the increase in the sodium reabsorption is mediated by two mechanisms. First, a direct effect of the angiotensin II on the kidney tubules. So the angiotensin II itself enhances the kidney tubules to reabsorb the sodium, and indirectly by stimulating the release of aldosterone from the adrenal cortex. So angiotensin II goes to the adrenal cortex, stimulating it to release aldosterone. And aldosterone effect, if you remember, was to move sodium back from my kidney tubules to the blood. Especially the distal convoluted tubules and the collecting ducts. Yeah. Remember, as you are moving the sodium back, this will allow you to pull water along with it. So water will follow the sodium by osmosis. As I recapture water and sodium that was about to be released in the urine, move them back to my blood, this will increase my blood volume back to normal, and so my blood pressure goes up back to normal. So recap here, we have detected a decrease in the blood pressure in the kidney by the granular cells in the juxtaglomerular apparatus that act as mechanoreceptors located in the afferent arterioles. So once they detect a reduction in the degree of stretch of the afferent arterioles, this means I'm not having enough blood coming in. This means for me, I have a low blood pressure. So the cells will be activated to release renin. Renin converts angiotensinogens that is normally circulating in your blood into angiotensin 1. Angiotensin 1 gets converted into angiotensin 2 by the angiotensin converting enzyme located in the capillary endothelium. And now looking at the effects of angiotensin 2. Angiotensin 2 first effect is going to be to constrict the arterioles, which will increase my total peripheral resistance, and so my blood pressure will go up. Two effects on the hypothalamus. First, stimulation of the hypothalamic thirst center and enhancing the hypothalamus to release ADH, which allows me to retain water. And as I retain water or add water by drinking a beverage, this will increase my blood volume back to normal, and so my blood pressure goes back to normal. The other effect of angiotensin 2 was to enhance the sodium reabsorption. And again, it acts by itself to increase, but this is a minimal effect. to increase the sodium reabsorption in the kidney tubules, but the more effective way by which angiotensin 2 mediates the sodium reabsorption is by stimulating the release of aldosterone. Aldosterone, when released, it's going to help me get the sodium back from the kidney tubules to the blood. And as I move the sodium, water will follow by osmosis. So as I get the sodium back to my blood, this will increase my blood volume back to normal, and so my blood pressure goes back up to normal. Another effect of angiotensin 2 is going to be by constriction the efferent arteriole in the nephron. If you remember, this is my Bowman's capsule. The blood comes in through an afferent arteriole to be delivered to the glomerulus. This is where I have the filtration taking place. And the blood that remains after I filter the blood coming in is going to be drained out from the glomerulus through the efferent arteriole. Then I have the proximal and distal convoluted tubules and loop of Henle. We'll draw this as just one tube like this. The efferent arteriole gonna keep forming capillaries around the kidney tubules, and we call those are my peritubular capillaries. If you remember, due to the difference in the diameter between the afferent arteriole and the efferent arteriole, normally I create a high hydrostatic pressure within the glomerulus, which allows me to drain the fluids out from the glomerulus into the Bowman's capsule. But remember, around, in some cases, more than 99% of what I filtered gets reabsorbed back from my kidney tubules to my peritubular capillaries. All right, so this is what normally happens. So if I do have a low blood pressure, how would I, when I constrict the efferent arteriole, this would help me increase my blood pressure back to normal. When I constrict the efferent arteriole here, I will be, as a result, reducing the blood pressure going into my peritubular capillaries. And as I reduce the hydrostatic pressure of the blood located here in my peritubular capillaries, this will allow more water to move back from the kidney tubules as they are facing a lower pressure, a lower resistance for this water to move back to my peritubular capillaries. And as a result, this will increase my water reabsorption. And as I get more water back from my kidney tubules to my peritubular capillaries, this will increase my blood volume as I retain more water, and as a result, my blood pressure will go back up, back to normal. So this is again the other effect of angiotensin II, by which it helps increase your blood pressure back to normal in case of a decline. The last effect of angiotensin II by which it increases the blood pressure was by contracting the extraglomerular mesangial cells. If you remember, extraglomerular mesangial cells are located next to the glomerulus. And this is how the blood is coming in and out from the glomerulus. This is my Bowman's capsule here. This is my glomerulus, my glomerular capillaris. The blood is coming in through the afferent arteriole, and it's drained out from the glomerulus through the efferent arteriole. This is where I've got my extraglomerular mesangial cells. You see all the surface of the capillaries in contact with the Bowman's capsule. This is the available surface for filtration to take place. This is where I can allow fluids to travel from the glomerular capillaries into the Bowman's capsule. So all this is a surface that I would be able to use to allow the filtration to take place. When I contract my extraglomerular mesangial cells, I will be pulling the capillaries of the glomerulus away from the Bowman's capsule. You see on here, this is again my Bowman's capsule. And now, after the contraction of the extraglomerular mesangial cells, this is how the capillaries are going to be moving closer to one another. I contracted the extraglomerular mesangial cells here in between the glomerular capillaries. And now the surface area available for the filtration to take place after the contracting the extraglomerular mesangial cells has dramatically been reduced. So this will allow me to filter less blood. So if I have a decrease in the filtration, so this means I have water retention. So preventing water from like kinking a water hose. Exactly, Dennis. I'm retaining water, and if I'm retaining water, this means I am increasing my blood volume because I didn't lose this water out, so it's kept as blood volume. And if I have an increase in my blood volume to normal, I have an increase in my blood pressure back to normal. This is what we have seen and discussed concerning how the kidney is responding to regulate the blood pressure. Again, the kidney responds to a decline in the blood pressure by increasing it. How detects it? Granular cells of the juxtaglomerular apparatus will detect a decrease in the degree of stretch. They respond by releasing renin that converts angiotensinogen to angiotensin 1. Angiotensin 1 gets converted in the endothelial capillaries by the endothelium of the capillaries into angiotensin 2 by an enzyme called angiotensin converting enzyme. Angiotensin 2 will perform a vasoconstriction of the arterioles, which stimulates the hypothalamic thyroid center and stimulates the hypothalamus to release ADH, increase sodium reabsorption directly and by stimulating the release of aldosterone. Yes, we need to know all those points for the essay question. Stimulating the release of aldosterone from the adrenal cortex, and the another effect of angiotensin 2 will be to constrict the efferent arteriole, which allows me to get more water reabsorbed back to my blood. Last effect by which angiotensin 2 increases the blood pressure is by contracting the extraglomerular mesangial cells, which reduces the filtration surface area and thus retaining water in my circulation. All right, so the second component that we have discussed was how the heart is going to be regulating the blood pressure. Remember, always remember that the heart hates high blood pressure, and whenever it detects it, it's going to be trying to reduce it back to normal. So the heart, simply speaking, is counteracting the effects of the kidney to increase the blood pressure by canceling its effect or the effects of angiotensin 2 mainly. How it detects it? It detects it by detecting the degree of stretch. So if I have a high blood pressure, there will be an increase in the degree of stretch applied to the atrium. And once the atrium detects that there is an increase in the degree of stretch because of the increased volume of blood coming back into the heart, once I detect that there is an increase in the degree of stretch in the atrium starts to release a hormone called the atrial natriuretic peptide. Atrial natriuretic peptide is going to be acting on 3 target organs. Preventing them from releasing their hormones. So the first target organ is going to be the kidney, to be more specific, the granular cells of the juxtaglomerular apparatus. It will prevent the kidney's granular cells from releasing renin. And if I don't have renin, remember renin is responsible for the conversion of angiotensinogen into angiotensin 1 that gets converted into angiotensin 2. So if you don't have renin, you don't have angiotensin 2. And if you don't have angiotensin 2, you don't have vasoconstriction. If you don't have vasoconstriction, you have a vasodilation. And vasodilation means what for me? Means a decrease in the degree of resistance. And once I have a decrease in the resistance, in the peripheral resistance, this will result in a decrease in my blood pressure. The second target organ on which atrial natriuretic peptide is going to be acting is going to be the hypothalamus and posterior pituitary gland. by preventing the release of ADH, antidiuretic hormone. And if I don't have ADH, as we just mentioned, ADH was doing what? It was inserting the aquaporine channels in the collecting tubules to move water back to the circulation. So here, when I'm not having ADH, I don't have that water retention that was allowed by ADH. And if I don't have that water retention, I have water loss. And if I have water loss, this means what for me? This means a decrease in my blood volume, which will result in a decrease in my blood pressure. The third target organ for ANP, adrenal peptide, was, if you remember, the adrenal cortex. And it acts on the adrenal cortex to prevent the release of aldosterone. And if I'm preventing aldosterone release, this means I'm not moving the sodium back, I'm preventing the sodium reabsorption. And if I'm preventing the sodium reabsorption, the sodium and the water that was following it will be lost out in the urine. So I will have a sodium and water loss. This results in a decrease in my blood volume, which ends up reducing my blood pressure. All right, so those are, simply speaking, the two components of the essay question. So everything that I've just mentioned needs to be stated in your response according to what's being asked. All right. Questions? Any question related to the essay question here? The order, no. It doesn't matter which order you go with in terms of those 5 effects of angiotensin II. But you need to be clear in the description of the sequence of events, like detecting the decreases of blood pressure, stimulates the granular cells, releasing renin, angiotensin to angiotensin 1. So those, the order here needs to be maintained in the description for it to make sense for somebody who's reading your response. But the order in terms of points, those 5 points, it doesn't matter the order, although vasoconstriction is going to be a very first thing as an effect of angiotensin II, and then you can hypothalamus effect and sodium reabsorption, those on the same level, and then those constriction of the efferent arteriole and contraction of the extraglomerular mesangial cells are on the same level as well. So I would highly recommend you to start with vasoconstriction, although this will not affect the grade in any way if you put them in any order. but I highly recommend you to start with vasoconstriction of the artery wall as a very first response. Actually, this is where the hormone got its name. It's called angiotensin. It constricts tension. It puts tension on the angio blood vessel. All right, so moving on to the diagrams. A quick overview of the important diagrams. We don't have any diagrams that are important for our lab exam from the body fluids chapter. Again, as what we're used to, we're going to be rating the diagrams out of 5 stars, 5 stars being a high priority. And 1 star will be a low priority. Low priority means you spend less time studying the diagram. High priority means you need to spend more time studying the diagram. So the first diagram here is a 4 stars diagram. It's showing the different components of the urinary system. Here we're looking at the kidneys, the ureters, the urinary bladder, and the urethra. Important organs around the kidneys that are related to the kidneys here in the abdominal cavity. We can see the major artery and vein, abdominal aorta here, the inferior vena cava, and the direct branches that are coming from those 2 large vessels to and from the kidney, the renal arteries and renal veins. And then we do have some branches on here. If you remember, the branches from the abdominal aorta, those are my common iliac arteries, and the 2 branches of the inferior vena cava, those are the common iliac veins. Some relationship to the urinary bladder, as you see on here, just behind the urinary bladder and the urethra, we've got the uterus, and behind the uterus is going to be the rectum. This is again a 5 stars diagram. 5 stars diagram. 4 stars, I'm sorry, 4 stars diagram. A transverse section, a diagram shows the transverse section of the abdominal cavity, in which I've removed all the abdominal organs located in the peritoneal cavity. So what's remaining are the kidneys and the major vessels behind the peritoneal cavity. Behind the peritoneal cavity, retroperitoneal. So this is a 3 stars diagram. Remember, this is the peritoneal cavity and not the abdominal cavity. A lot of people answer this as abdominal cavity on the exam. This is just the peritoneal cavity because everything on here is the abdominal cavity, including the kidneys, abdominal aorta, and inferior vena cava. The space surrounded by the peritoneum is not the abdominal cavity, it is the peritoneal cavity. We can see the kidneys, fibrous capsule of the kidney, the surrounding fat to support the kidney called perirenal fat capsule. The level at which we've got this section at is the 2nd lumbar vertebra. This is where we've got the transverse section of the abdomen. A 10 stars diagram, either diagram may appear on your test. So you need to be familiar with both of them. This is a 10 stars means it has to be on your test. It's a must be on your test. As we've learned, the kidney has 2 main components, an outer layer, this is my renal cortex, and an inner layer, this is my renal medulla. At the junction between the renal cortex and medulla, we've got the triangular shaped structures on here. Those are my renal pyramids. The tip of the pyramid is what we call the renal papilla or the papilla of the pyramid, draining the urine from the renal papillae. So any tube that is directly connected to the renal papillae, to the papilla of the pyramid, this is going to be a minor calyx. So getting the urine drained into small ducts directly from the renal papilla, those are going to be my minor calyces. Multiple minor calyces or a number of minor calyces will drain into a larger duct. This is going to be called a major calyx. So those are minor calyces. Those are my major calyces. And the major calyces will drain into a larger funnel-shaped tube. This is going to be my renal pelvis that will drain the urine down through the urethra. This is the hilum here, and the artery coming in is my renal artery, and the vein draining the blood out is going to be my renal vein. Those regions between the adjacent renal pyramids, those are my renal columns. Renal columns are those regions located between the adjacent renal pyramids. A must be on your test diagram, a 10 stars diagram, very important, along with the other diagrams describing the nephron. We can see here the different kidney tubules forming my nephron. We've got first the Bowman's capsule, proximal convoluted tubules, loop of Henle, distal convoluted tubules, and finally collecting ducts. The artery bringing the blood in. This is my afferent arteriole. The blood getting the blood out from the glomerulus. This is my efferent arteriole. A more complicated, but again, a must be on your test is going to be the diagram showing the two different types of nephrons. If you remember, you have 1 million nephrons per kidney. We do have two types, cortical type and juxtamedullary nephrons type. Cortical nephrons, if you remember, this represents most of the nephrons in the kidney. They are having a short loop of Henle that travels for a brief depth into the medulla. Compared to the juxtamedullary nephrons, they do have a long loop of Henle surrounded by those straight vessels, the vasa recta. If the blood is coming from an artery, there's going to be an afferent arteriole. Coming out from the glomerulus, this is going to be the efferent arteriole. A way for you to be able to tell whether you're looking at proximal convoluted tubules or distal convoluted tubules is to look at the loop of Henle. The loop of Henle has a thin segment and a thick segment. The thin segment, if you see on here, this is part of my descending limb of the loop of Henle. So the descending limb means I am connected to the proximal convoluted tubules. So this tells me that this tube on here is going to be the proximal convoluted tubule. And this one on here, that is a continuation of the thick segment of the loop of Henle. This is going to be my distal convoluted tubule. Blood coming in will travel through the arcuate artery that is separating the adrenal cortex from the, the renal cortex from the renal medulla. And the blood travels into the cortex, passing through the radiate artery. So you travel from the arcuate artery to the radiate artery, and from the radiate artery, you're going to be distributing the blood to the glomeruli of the nephrons in the cortex through the afferent arterioles. And if you remember, again, the efferent arteriole is called an arteriole because the blood is going to keep moving away from the heart, and the blood here is going to be traveling around my proximal and distal convoluted tubules. This is going to be my proximal, my peritubular capillaries, and it also can form the vessels around the long loops of Henle. This is what we called the straight vessel vasa recta. All right. Any question here? Any question here? For Venetia, yes. So the essay question is the one that I will be manually grading for this lab exam. Yes. So I will be grading them after the exam is closed. Any question? Any question? All right. So moving on here. Another diagram is a 3 stars diagram. This is a simplistic diagram showing the different components of the kidney tubules and the associated vessels, along with those 3 arrows are showing the 3 steps of the urine formation. So this is really important to remember that the very first step of the urine formation is the glomerular filtration. Second is the tubular reabsorption, and the third is going to be the tubular secretion. A must be on your test diagram is going to be the diagram showing the structure of the urinary bladder. You can see the ureters on here are going to be collecting the urine to be drained into the urinary bladder through those openings on the dorsal wall of the bladder. Those are the ureteric orifices. We can see that the urinary bladder is located outside of the peritoneal cavity. So this is my peritoneum on here, just covering one side because the bladder is outside of the peritoneal cavity. It's not surrounded by the peritoneal cavity. We can see the muscle on here, this thick layer of smooth muscles located in the wall. So we call this muscle is my detrusor muscle. And on the inside, we can see those ridges in the mucosal lining of the urinary bladder. We call this as my rugae. We see a triangular shaped funnel that will be collecting the urine to be directed towards my urethra. This is called the trigone. And something I want you to notice on here is, see, this is my trigone, this triangular shaped part. And this is the urethra, the tube. The connection here between the trigone and the urethra, this is the bladder neck. Again, this is the bladder neck on here, because on the exam, some people will call the trigone the bladder neck. Some people will call the urethra the bladder neck. The bladder neck is just this point. This is the junction between the urethra here and the urinary bladder. This is again my bladder neck. We can see two muscles, two distinctive muscles. I can see muscle on here around the bladder neck. This is gonna be, see this line that demarcates the muscle. This is my internal urethral sphincter. This is inside of the urethra. And one located on the outside of the urethra. This is the external urethral sphincter. See, this is my external urethral sphincter. This is my internal urethral sphincter. The most common wrong answer I usually see as when you identify the external urethral sphincter, you state urogenital diaphragm. Urogenital diaphragm is the whole pelvic floor, the whole thing. It has many other muscles other than this specific one. So yes, the external urethral sphincter is part of the urogenital diaphragm, but if I'm marking this specific one, you need to be as specific as possible. So this is going to be the external urethral sphincter. How about the rest of it? The rest of the muscles around it? Yeah, we didn't study them in details in this chapter, so for us, a broader identifying name can be considered, because those are different muscles that will have different names. But collectively, they are called the urogenital diaphragm. Questions. Here, why those are circled? Because those are the three types of muscles that will regulate the micturition process. This doesn't mean that those are the only three that you need to know. You need to know all the different markings on the diagram. All right. Questions? Any question? So looking on here, it's essentially the same diagram, the same previous diagram, but it's showing the difference between the female urethra and the male urethra. If we're looking on here, the male urethra is much longer. That's why it is less common for males to develop UTIs compared to females. The first portion of the urethra here is traveling through the prostate glands. So we call this portion is the prostatic urethra. And then I have a portion of the urethra that travels through the urogenital diaphragm. This is called the intermediate urethra or membranous urethra. And then we have this part of the urethra that travels in the penis. This is going to be the spongy urethra or the penile urethra. Spongy urethra or penile urethra, those are essentially the same thing. Spongy urethra or penile urethra. The opening of the urethra in either male or female is going to be the external urethral orifice. So I want you to notice the difference between the urethral orifice, the opening of the urethra, and the ureteric orifices. Ureteric, those are of the ureters. Urethral, those are for the urethra. So don't mix urethral with ureteric. All right, so why is this part of the urethra is called spongy urethra, the part that travels in the penis? I understand it's called also penile urethra because it travels in the penis, but why spongy urethra? Because when we study this in the reproductive system, we're gonna learn that the penis has 3 erectile tubes. This is a transverse section in the penis here. We have 3 erectile tubes. The 2 on the dorsum of the penis, those are the corpora cavernosa, and the 1 on the ventral surface through which the urethra is traveling is called the corpus spongiosum. Corpus body, and spongiosum is like sponge. It is compressible. This is where the urethra is going to be traveling. So that's why it got its name, spongy urethra. Spongy urethra. All right. All the rest of the markings are actually repeated from the previous diagram. Which one is more susceptible to develop a UTI? Again, is going to be females. Because of the shorter urethra, the bacteria doesn't have to travel for a long distance facing the immune system until it gets to the urinary bladder for it to have a better proliferation. Our last diagram here is a must be on your test diagram. This is a 10 stars diagram. This is a histological picture. I would rate it 10 stars. This is a must be on your test. Histological picture showing the different components of the nephron. The very first thing I can see on here is going to be the cluster of capillaries surrounded by this white region. Those capillaries are my glomerular capillaries, or the glomerulus. And this empty space is the empty space inside the Bowman's capsule, or the glomerular capsule. So you have the glomerulus on the inside surrounded by the Bowman's capsule. This is what we have been drawing all the time here. This is the glomerulus. This is the Bowman's capsule around it. This is what they do actually look like. All right, this is what we have been drawing. Glomerulus and Bowman's capsule. This is actually what they look like. And we see tubules scattered around everywhere. 2 main types of tubules that you can see on here. Those are the proximal convoluted tubules and the distal convoluted tubules. The question is, how would I differentiate between the proximal convoluted tubules and the distal convoluted tubules? The proximal convoluted tubules, if you remember, they do perform lots of reabsorption. That's why they do have dense microvilli. So the cells in the tubules, lining the tubules, are gonna carry a lot of microvilli to allow higher levels of reabsorption. Compared to the distal convoluted tubules, they do have much less microvilli because they are performing mainly secretion, not reabsorption. So they do perform some reabsorption, but mainly they are going to be managing secretion. So how this will help us differentiate between the tubes on the slide on here. If I'm looking at the tube, this is a tube on here. And I see that this tube has a fuzzy lumen. Fuzzy lumen. If I have a tube that has a fuzzy lumen, this tells me it's lined with cells that do have dense microvilli. So the tubes with fuzzy lumens, those are going to be the proximal convoluted tubules. And the tubules that will have clear lumens, so a clear lumen on here, clear, it's not fuzzy. So this tells me that those tubes are lined with cells with less microvilli. So they don't have that much microvilli. That's why the lumen inside the tube is clear. See how is it different between the lumen of this one and the lumen of this one. So this has a fuzzy lumen, this has a clear lumen. Which one will have a fuzzy lumen again? The one that has microvilli, lots of microvilli. Those are my proximal convoluted tubules. And the ones with clear lumens, those are the ones with less microvilli, and those are the distal convoluted tubules. So those are pretty much all the diagrams that you need to remember for this exam. So again, here one last recap. This diagram is a 4 stars diagram. This one is a 3 stars diagram. Must be on your test diagram, either one of them. 10 stars. Another 10 stars diagram showing the simplistic structure of the nephron, the kidney tubules mainly. Another highly important diagram is a 5 stars diagram showing the different components of the nephrons and the surrounding vessels, as well as showing you the difference between the two types of the nephrons. A relatively moderately important diagram here is the 3 stars diagram. Remember, you need to be able to tell the 3 different steps of the urine formation. It must be on your test diagram showing the structure of the urinary bladder and its association with the ureters and urethra. Must be on your test. A highly important one that actually going to be repeating all the different components stated in the previous diagram with the added structures of the Mediolith, the different parts of the Mediolith. Here, this is the only added information from the previous slide. This is a 5 stars diagram. And the last one on here is the histological picture of the different components of the nephron. We need to be able to identify the glomerulus, Bowman's capsule, or the glomerular capsule, distal convoluted tubules, which will have clear lumens, the proximal convoluted tubules, which will have fuzzy lumens. All right, any questions? Any questions? questions.